It's 1959, and Stanley Cohen is pacing the halls outside his lab, as he often does when he's trying to think. He puffs on a corncob pipe, its frequent usage having burnt holes in his pants pockets. Stanley is trying to figure out a problem. He recently stumbled across a curious side effect in his research. An extract meant to stimulate nerve growth has instead affected the eyes and teeth of newborn mice. Was some new biochemical responsible? And if so, what exactly was it doing? Biology and health were not just fields of study for Stanley. They had been a part of his life since childhood. Born in Brooklyn in 1922 to Russian Jewish immigrants, Stanley had been stricken with polio as a child, and after recovering from the disease, was left with a limp for the remainder of his life. His family was of modest means. Stanley's father, a tailor, and his mother, a homemaker, had received limited educations themselves, but the two of them made sure to teach Stanley the value of intellectual curiosity. Out of this humble upbringing, Stanley managed to excel in high school and was subsequently accepted to Brooklyn College. Fortunately for me, my college education was most thorough, he wrote in his Nobel autobiography. Perhaps equally important, he added, was the fact that Brooklyn College was a city school and had a policy of no tuition. The cost of an education would have been prohibitive for my parents. It was at Brooklyn College where Stanley first became interested in how living things grow from the tiniest of structures to complex organisms, and he focused his studies on the biology of cells and embryonic development. He felt that the key to understanding these phenomena would likely be found in the application of chemistry to biology, and consequently decided to major in both fields. In 1943, Stanley graduated from Brooklyn College and worked briefly as a bacteriologist in a milk processing plant. Combining the money from his earnings with fellowship awards, Stanley was able to attend graduate school at Oberlin College, where he received a master's degree in zoology, and the University of Michigan, where he pursued his PhD. His thesis was on nitrogen metabolism in earthworms, a job that required him to spend many nights collecting thousands of worms around campus. But the dirty work brought its rewards, and in 1948 Stanley earned his PhD as well as a job working with far more precious subjects, premature infants at the University of Colorado's Pediatrics Department. In 1952, Stanley felt compelled to explore the newly emerging application of radioisotopes to biology, a field then being pioneered by fellow CUNY graduate and future Nobel laureate Rosalind Yalow, among others. Consequently, he took a job at Washington University in St. Louis, where he learned how to use radioisotopes in the study of animal eggs and embryos. He also gained valuable experience studying and discussing research journals with another future CUNY laureate, Arthur Kornberg. But it was a different colleague at Washington University who helped put Stanley on the path that would define his career. Rita Levi-Montalcini, an Italian Jewish scientist who had spent World War II studying nerve cells in secret under the Nazi occupation, had come to Washington University after the war to continue her work. While there, she discovered a chemical in mouse tumors that appeared to stimulate nerve growth. It was a protein that came to be known as nerve growth factor, or NGF. Stanley's background in chemistry made him the perfect candidate to further explore this new discovery, and in 1953, Rita asked Stanley to help her isolate NGF in the lab. "I did the chemistry, she did the biology," Stanley told the Washington Post in a 1986 interview. It was a completely collaborative effort. Together, the two scientists were able to isolate NGF and determine its chemical structure. It was the first time a biochemical compound controlling cell growth had been isolated. But it wasn't the last. Soon after, Stanley would make a discovery of his own, and entirely by accident. While studying NGF in the salivary glands of mice, Stanley had injected salivary extracts into their newborns. He noticed that the injections caused the newborn mice to open their eyes about a week earlier than normal. He also noticed that their teeth appeared to grow more quickly. But NGF wasn't the cause. Injecting the nerve growth factor by itself had no such side effects. Stanley realized that the salivary extracts must contain something else. In 1959, Stanley Cohen took a job as an assistant professor at Vanderbilt University, where he decided to focus his research on this accidental discovery. But not everyone believed it was worth his time. People used to sort of laugh when he started talking about something that caused mice to open their eyes, a colleague later told the New York Times in an interview. But Stan has always been very focused on what he wanted to know. Assisted by only one technician and one postdoctoral student, Stanley was able to isolate the mystery substance. It was a protein similar to NGF, but one which stimulated growth in epidermal cells rather than nerve cells. Stanley and his colleagues dubbed the new substance EGF, epidermal growth factor. Over the subsequent decade, improving technology allowed Stanley to determine the chemical structure of EGF and to isolate it in humans. And after many more days of pacing outside his office and devising a variety of experiments, Stanley was eventually able to identify the cell receptors that attach to epidermal growth factor, as well as a mechanism by which EGF enters a cell and causes it to divide. Initially, the growth factors discovered by Stanley and Rita were met with skepticism by scientists who believed them to be of little consequence. But as Stanley and others demonstrated more about how they worked, their importance became impossible to overstate. EGF was found to have applications for wound healing, skin transplants, and for heart, liver, and kidney disease. Researchers also found that targeting EGF receptors with drugs could help treat a wide variety of cancers. In particular, lung cancer tumors were found to have mutations in their EGF receptors, and targeting those mutations could lead to dramatic recoveries that one medical professional described in 2011 as Lazarus-like. Stanley Cohen spent the remainder of his career studying EGF and other growth factors, and after retiring, continued to channel his passion for biochemistry into the next generation, mentoring elementary and high school students in science. Many new things are found by accident, he told a group of students in 2007. If you're prepared to see the accident, you can find it. In 1986, Stanley Cohen, the soft-spoken, irrepressible optimist who liked to smoke corncob pipes and play the clarinet in his free time, was awarded the Nobel Prize in Physiology or